Hello everyone! Today we are visiting the Lab at the TU Vienna, which is the Micromechanics Lab, to check out the CortMech project of Martin Frank, which is conducting the project together with me. So let's have a look. Martin!
Hi Andreas!
Hi, nice to meet you!
Hi, nice to meet you!
Okay, what is this CortMech project about? So basically it is about the mechanical properties of cortical bone. And why is this interesting? Because the mechanical properties of bone allows us to do what we do on a daily basis, standing, running, jumping, so everything we are doing in our lives. And there are diseases that corrupt these properties, like osteoporosis, where, for example, the fracture load of certain bones are decreased. And we are investigating how the mechanical properties of bone are changing, and for that we need to measure them. In a previous project we focused on the inside of these joint regions. We investigated the properties of single trabecular, depending if they are healthy or osteoporotic. And in this project right now, where we show you what is going on in a few minutes, we are focusing on cortical bone. That means we are cutting out tiny specimens of the cortical region in order to test them mechanically and to extract material properties. So where are we taking our samples precisely, Martin?
Okay, yeah, thank you Andreas. So the samples that you will see we're milling out will be extracted from the diaphysis, because here we have only cortical bone. This is rather thick, it's around 10 millimeters, so there it's quite straightforward to dissect the cortical bone specimens. And you can imagine, since bone is loaded in this direction, the osteons are orientated longitudinally also in this axis, so you can imagine small cylinders in that direction, and for that reason you will get a different behavior if you mill out the samples in this direction or transversally, like if you cut through the cylinders. Mm-hmm. But as Andreas has mentioned before, in osteoporosis the fracture usually occurs in the femoral neck, because here the cortex is much thinner, and here you will the bone will fracture if you have a fall on the side, for example. For that reason, we will extract them in the next step of our study, very tiny specimens from that region, and then we will investigate the mechanical properties and determine if they are different in osteoporotic bones in comparison to healthy bones.
So, okay, Mr. Frank, this is a special freezer, which is not at -20 as I can see here, which is colder.
-80 degrees.
So why are the bones stored at -80 degrees?
To actually they are preserved better, that we do not have much degradation of the biological tissues actually. Yeah, this is now the proximal part of the human femur, and here you see we cut the neck of the femur with the head. So here we only have the trochanter major and minor, but we do not have the head anymore because this was used in another study. So here we have the diaphysis, which was used in our current project. And yeah, we cut here then slices, actually with a saw nearby. And then we're able to extract long, thin rectangular pieces, which we slice then into small, very thin, it's like 300 micrometer thick bone slices.
So these are now thin slices of cortical bone that are stored here in buffered solution, and that will be now cut to shape on the CNC router.
Okay, then I will place our sample here, and we'll try to align it perpendicular
to the machine axis.
To the machine axis. Mm-hmm. Yeah. that
the machine is cutting it.
The machine is cutting it parallel to the osteonal orientation actually. And we have to clamp it to avoid, of course, that the sample is moving during the milling process. Now,
It should be tight, you cannot move it anymore.
yeah.
Yes, we are applying water to cool the milling process.
Okay, we've already set now the zero position, so this is exactly the center of our sample. We've loaded the shape that we're going to mill in a software, and now we can start the milling process.
We're taking out the sample.
Yeah, and here you see the finished milled sample, and yeah,
Of cortical human bone.
cortical human bone. And now we're going to inspect it in the microscope. You can nicely see the longitudinal orientation now of the osteons. So if you have a look on your own.
You see lines going from left to right. These are the Haversian channels that enter and exit the surface of the sample. Okay, let's check if the sample will fit into the sample holder.
Here, yeah, and we are lucky, it fits nicely into the sample holder. So yeah,
And these two sample holders go into the testing machine and will pull or push on the sample.
I will show you now the last step that we need for our sample preparation before we can do the actual mechanical tests. So in this step we will apply now a speckle pattern on our small tensile specimens in order to perform optical strain tracking directly on the samples. So we see here our samples placed under the microscope, and here you see now our sample aligned. We added now the masks, and here you can see now these 5 lines which are spaced 1 mm apart to allow the optical strain tracking. In order to hold the mask in place, I added now a lid, and now the sample is ready to be speckle patterned. And here we go now to our box where we apply the speckle pattern. Here you see our mounted samples, and we use a conventional black spray pattern. And we spray now into the box, not directly onto the samples, but beneath, to have kind of very small particles which can settle down then onto the samples in between the stripes here. We will have a look, and you see that everything is now spattled, and we will take a detailed look in the microscope. Here you can see now our speckled samples, and if we zoom in, you can see that we have now the speckle pattern on the masks and of course directly on the bone. If we remove now the mask, you can see nicely that we have here our applied speckle pattern, and here we can zoom in again, and you see now we have these stripes which are separated 1 mm apart. Our samples are now ready to be rehydrated in a saline buffer solution again, and then they are ready for testing.
Okay, so the samples are now prepared. We have them here in the buffered solution, and now they are going to be tested mechanically under tension. And for doing this, we have our test equipment here, which is consisting of this load frame. This is the gray part here, and this load frame is doing something very simple. So it's only moving this rod in there up and down. And for measuring force, we have mounted this 100 Newton load cell here, which is this aluminium frame with this funny hole. And at the bottom we see the sample holder itself. So it's a bit complicated, and we will give more detailed shots later. But the samples are going to be mounted in these grooves here. And the bottom part, the bottom black part, is mounted at the bottom of the load frame. So when this load cell and rod is moving upwards, we can apply tension on the sample. And now the question is how deformation is measured in this test setup. In this case, as you remember, we sprayed on these very precise and slim lines on the sample, and we are tracking the deformation of those lines with this video microscope. You see on the right side currently the blurry image of the video microscope. One more thing, at the bottom here you see the load profile, which is actually not force but prescribed in terms of displacement. So we have a cyclic loading here, and at the end we pull the sample until fracture.
Okay, so I take now the sample and I place it here, and now I align it into the sample holder on the bottom and on the top. Now I can add the spacer to apply moderate pressure then with the clamps to have the sample in place. Clamp the sample. We can approach now to 0.2 Newton.
So we're ensuring now that the sample is aligned by reaching 0.2 newtons, and then now it's stopping. Okay.
Yeah. Now we can clamp it a little bit more tightly, not very tight, because then the sample might be damaged, but still tight enough that movement out of plane is avoided. Okay. Now the sample is fixed in the sample holder, and we pour in actually now our saline buffer solution, that the sample stays hydrated during testing.
And we see very nicely the sprayed on speckle patterns here.
And now we're ready to start the test. So as we've explained before, we apply now our cyclic loading profile. So I set the displacement now to 0. And you see that we already have a preload applied here. And now we start with this loading profile. We can start our test. And we also start here now the video frame. And now we have an initial 0 displacement, and then now you see the displacement increases and the sample starts deforming. Here you see now the finished video, and I will play it faster. So you see now here the movement up and down also of the sample. And when we come to this point, you see here that we have a so-called whitening effect in this region and in that region. So there apparently damage accumulates, and this is the reason why light refraction is changed. And when we unload again, this effect is diminished because the cracks close. But when we load again, you see now that here we have these regions which accumulate lots of damage. Then we unload again, and when we do this now the third time, we see that first here a crack starts, but then very rapidly you see that it fractures actually here on the top, but localized in 2 regions, whereas here we have almost no deformation or very small deformation.
Let's have a closer look at the results after data evaluation. The force data could be directly extracted from the load cell data, but the displacement, which is the change in length of the measurement distance, has to be extracted out of the video feed. And this is what you see here. So the points of the speckle pattern are tracked and averaged as the two lines at the top and at the bottom, and the distance between these two lines represents our displacement measure. This is the end result for this particular sample, a force displacement plot. You see a linear range up to 18 newtons, then yielding is occurring up to a maximum load of around 27 newtons. Then, beginning at 60 microns, we have a softening behavior till 90 microns displacement, where the sample was fracturing. The curve is quite noisy, which is a result of the very small displacements that had to be tracked. That was the experimental procedure of the CortMech project. I hope it was interesting, and have fun with mechanical testing. Goodbye.
Yeah, also I hope too that it was interesting for you to see how we actually do all of these micromechanical stuff here in the lab, and have fun in future and enjoy micromechanics.
Bye.
Bye.